The powerhouse of the cell, a deep dive into mitochondrial function. The mitochondria are often famously relegated to a single, catchy tagline, the powerhouse of the cell. While that metaphor is catchy and technically accurate, it barely scratches the surface of what these complex double-membranous organelles actually do for eukaryotic life. To understand the function of mitochondria is to understand the very chemical engine that drives biological existence. As of 2026, our understanding of these organelles has expanded from simple energy producers to sophisticated signaling hubs, regulators of cell death, and key players in the aging process and metabolic health. The architecture of energy. To grasp the function, one must first look at the form. Mitochondria are unique among organelles because they possess their own genome mtDNA and a double membrane structure. The outer membrane is smooth and serves as a gateway, while the inner membrane is deeply folded into structures called crista. These folds are not just for show. They maximize the surface area available for chemical reactions. Within this inner membrane lies the matrix, a dense soup of enzymes, ribosomes, and mitochondrial DNA. This structural compartmentalization is essential for the mitochondria's primary function: the production of adenosine triphosphate (ATP). The primary mission: cellular respiration and ATP production. The most critical function of the mitochondria is the extraction of energy from nutrients, primarily glucose and fatty acids, and its conversion into a usable chemical form. This process, known as cellular respiration, occurs in several distinct stages, most of which take place within the mitochondrial walls. The citric acid cycle, Krebs cycle. Once glucose is broken down into pyruvate in the cell's cytosol, it enters the mitochondrial matrix. Here, it is converted into acetyl-CoA, which enters the citric acid cycle. This cycle is a series of redox reactions that strip high-energy electrons from carbon-based molecules. These electrons are then loaded onto carrier molecules known as NAD and $FAD_2. While the cycle produces a small amount of ATP directly, its real value is in these electron carriers, the electron transport chain, etc., and oxidative phosphorylation. This is where the powerhouse earns its name. The NADH and $FAD_2 produced in the matrix travel to the inner mitochondrial membrane. They donate their electrons to a series of protein complexes known as the Electron Transport Chain. As electrons move through these complexes, the energy released is used to pump protons H+ from the matrix into the intermembrane space. This creates a massive electrochemical gradient, essentially a biological battery. The only way for these protons to flow back into the matrix is through a specialized enzyme called ATP Synthase. This protein acts like a molecular turbine. As protons flow through it, the enzyme rotates, providing the mechanical energy to phosphorylate adenosine diphosphate (ADP) into ATP. The final electron acceptor in this chain is oxygen, which combines with protons to form water H_2O. This is why we breathe, to provide the sink for the electrons that power our cells. Beyond ATP, the regulatory roles. If mitochondria only produced energy, they would still be the most important organelle in the cell. However, modern research shows they are also the command and control centers for several vital biological pathways. Calcium homeostasis. Mitochondria work in tandem with the endoplasmic reticulum to regulate the concentration of calcium ions $Ko2+ within the cell. Calcium is a universal signaling molecule used in muscle contraction, neurotransmitter release, and fertilization. By rapidly soaking up excess calcium and releasing it when needed, mitochondria act as a buffer, ensuring that cellular signals are precise and that the cell doesn't succumb to calcium overload, which can be toxic. Regulation of programmed cell death. Apoptosis. Mitochondria decide when a cell's time is up. When a cell is severely damaged or infected, the mitochondria release a protein called cytochrome C into the cytosol. This acts as a suicide signal, triggering a cascade of enzymes called caspases that systematically dismantle the cell from the inside out. This process, apoptosis, is vital for preventing cancer and ensuring proper embryonic development. Heat production thermogenesis. In specific tissues, such as brown adipose tissue, brown fat, mitochondria have a short-circuit protein called thermogenin, or uncoupling protein 1-UCP1. Instead of using the proton gradient to make ATP, UCP1 allows the protons to leak back into the matrix, releasing the energy as pure heat. This is how human infants and hibernating mammals maintain their body temperature in cold environments. The genetic frontier, mitochondrial DNA and inheritance. Mitochondria are the only organelles in animal cells that contain their own DNA. This DNA is circular, resembling bacterial DNA, which supports the endosymbiotic theory, the idea that mitochondria were once free-living bacteria that were engulfed by a larger cell billions of years ago. In humans, mitochondrial DNA is inherited almost exclusively from the mother. Because mtDNA has a high mutation rate and lacks the robust repair mechanisms of nuclear DNA, it is often used by scientists to trace maternal lineage back through thousands of years. Furthermore, mutations in mtDNA can lead to mitochondrial diseases, which often affect high-energy organs like the heart, brain, and muscles. Mitochondria in health and aging, as we look at the latest data from 2025 and 2026. The focus has shifted toward mitochondrial quality control. Our cells have a process called mitophagia, specialized form of autophagy, where the cell identifies, breaks down, and recycles damaged mitochondria. When mitophagy fails, broken mitochondria accumulate, leaking reactive oxygen species, ROS, or free radicals. These ROS cause oxidative stress, damaging DNA and proteins. This process is now recognized as a primary driver of aging and neurodegenerative diseases like Parkinson's and Alzheimer's. Current therapeutic research is heavily invested in finding ways to boost mitochondrial biogenesis, the creation of new mitochondria, and improve the efficiency of the electron transport chain to extend human health span. Summary of functions. The mitochondria perform a symphony of tasks that keep the organism viable. ATP. Production. The conversion of chemical energy from food into cellular fuel. Metabolic integration. Facilitating the breakdown of fats, beta oxidation, and the synthesis of heme used in hemoglobin. Cell signaling. Communicating with the nucleus to coordinate growth and adaptation. Apoptosis. Managing the life cycle and death of the cell. Ion regulation, maintaining the delicate balance of calcium and other electrolytes. The future of mitochondrial science. We are entering an era where mitochondrial transplants and mitochondrial DNA editing are no longer science fiction. By understanding that the mitochondria are not just batteries, but dynamic, communicating organisms within our cells, medicine is moving toward a more holistic view of metabolic health. Whether it is through exercise, which stimulates the growth of more crista and more, mitochondrial through targeted antioxidants, maintaining mitochondrial function is the key to vitality. Mitochondria are the bridge between the air we breathe, the food we eat, and every movement we make. They are the ancient guests that became the essential masters of the eukaryotic cell, proving that even at the microscopic level, life is a collaborative and incredibly high-energy endeavor.